Hi everyone, this is the second mini lecture in Chapter 11. In this mini lecture we will be covering infection to disease. First, I'd like to go over some vocabulary which will make this chapter a little bit more understandable. We want to start with the progress of infection and talk about what a pathogen is. So a pathogen is any microbe whose relationship with its host is parasitic and results in infection and disease. Now that type of infection or severity of infection depends on the pathogenicity of the organism and the condition of the host. So what is pathogenicity? That is an organism's potential to cause infection or disease. So how good it is at causing infection or disease. And we have different levels of pathogens. The first is considered a true pathogen, and this is a microbe that is capable of causing disease in a healthy person with normal immune defenses. So these are microbes who can cause disease even if our body is functioning perfectly normally. So they're generally associated with a specific and recognizable disease, and they can vary from severe to mild and even from severe to fatal. Examples of true pathogens would be the influenza virus, so perfectly healthy people can be infected by this, plague bacillus, and malarial protozoan. These are all microbes that can cause an infection in a healthy person with a healthy immune system. We also have pathogens which are considered opportunistic, taking advantage of an opportunity that has arisen. In this case, the disease may happen because the host's defenses are compromised, or if that pathogen becomes established in a part of the body that is not natural to that particular microbe. Our body may not then have a normal response. They generally are not considered pathogenic in a normal, healthy person, so they're really looking for an opportunity, something that has gone wrong in a healthy individual. They generally don't possess well-developed virulence properties, and some examples of this include the Pseudomonas species and Candida albicans, which is a yeast, and these are ones that actually need to have something change in a healthy host that open up an opportunity for infection. So what do we mean by virulence? Virulence of a microbe is determined by two things: its ability to establish itself in a host and then to cause damage. So it must be able to do both of these. The virulence factor is any characteristic or structure of that microbe that gives it the ability to establish itself in the host and cause damage. So let's talk about the 5 steps that we see with an infection or disease. Step 1, the microbe needs to become established, so it must enter the host in some way. So we have what is called the portal of entry. This is the route that a microbe takes to enter the tissues of the body to start an infection. And there's 2 different types of microbes we would be talking about. The first is exogenous. These are microbes that originate from a source outside of the body, so either from the environment, another person, or animal. We can also have endogenous microbes, and these are ones that already exist in the body, maybe part of a normal biota or a previously silent infection, that then establish themselves in the host. So step 2 is becoming established or attaching to the host. So we've already entered in step 1. Now we are looking at adhering to the host. So this is a process by which the microbes can gain a more stable foothold on host tissues. And this is dependent on binding between specific molecules on both the host and pathogen. So there's specific molecules that allow that pathogen to bind to the host. And the pathogen, therefore, is limited only to those cells or organisms to which it can bind. So there's specificity in this case for adhesion. And then firm attachment is almost always necessary for causing disease because the body is so good at flushing microbes from the tissues that it must have firm attachment to remain in the host. So that's step 2, adhesion. Step 3 in becoming established is surviving the host defenses. So particularly in humans, we have a very strong immune system. So the microbes that are not established as normal biota will likely encounter the host immune defenses when they first enter. They're going to be recognized as foreign to the body. And the general cell that we look at to recognize and to destroy the pathogen would be phagocytes. Phagocytes are cells that can engulf and destroy those pathogens by means of enzymes and antimicrobial chemicals. So in order to survive host defenses in step 3, the microbe must have some way of either evading the phagocytes or surviving that engulfment and not being destroyed by the phagocyte. So once we have that it has evaded the immune system, we have step 4, which is causing a disease. So how these virulence factors, these structures of the microbe that can contribute to tissue damage, start to cause disease in the host. And these virulence factors are adaptations that the microbe uses to establish itself in a host. There are 3 ways that a microbe can damage the host. First, it can directly damage the host through the actions of enzymes that it contains. Secondly, it can directly cause damage through the action of toxins, and these are both endotoxins and exotoxins that the microbe might produce. And then lastly, the microbe also can indirectly cause damage by inducing the host's defenses to respond too much, excessively or inappropriately to the infection. And this is where you would start to see disease caused. So the steps involved, there are 5 steps. We've talked about 4. I'll go over the 5th after this. But the first step that we have for a microbe causing disease in host is finding a portal of entry. And this could be the skin, GI tract, respiratory tract, urogenital tract, even the endogenous biota if something gets out of equilibrium. Next, that microbe must attach firmly, and this can be through the use of fimbriae, capsules, surface proteins, or viral spikes in the case of viruses. Then it must be able to survive those host defenses, so it must either be able to avoid phagocytosis, avoid death once it's been engulfed by the phagocytosis, or perhaps it's through the absence of a specific immunity for the microbe that has caused the infection. Next, it needs to be able to cause damage or disease, and it can do this through direct damage by toxins and or enzymes, or indirect damage, which can include that inappropriate excessive host response. And then the last part of this microbe causing disease in a host is the exit of the host. And there can be portals of exit, including the respiratory tract, perhaps in coughing, sneezing, salivary glands, skin cells, fecal matter, urogenital tract, or blood. So this is the 5th step, vacating the host, leaving the host once it has caused that disease. So there are specific avenues by which the pathogens exit. It's usually dependent on where their infection is. They can be shed through secretion, excretion, discharge, or even sloughed off tissue, for example, the skin. And if there are high numbers of the microbes in these materials, that it increases the likelihood that the pathogen then will go on to infect other hosts. And the portal of exit is often the same as the portal of entry, but some pathogens use a different route. And you can imagine with a GI tract infection, Often the portal of entry would be through ingestion. The portal of exit would be through excretion. So that is one that would be an exception to that. Hopefully this helps you understand the 5 steps of pathogens causing disease in hosts.